Okay, it's noon, so let's get started. Welcome to February's Statistical Seminar. Today we're going to talk about Bayesian analysis. Up to this point, all of the statistics that we've looked at have been from the frequentist viewpoint. But today we're going to look at a different perspective on how to do statistical analysis. So my outline here is I'm going to do a review from the frequentist or the classical perspective, which was the very first session I did back in October, I think it was. Then we will look at the Bayesian perspective. And as I thought about doing this, I thought, well, probably the best way to present Bayesian statistics is to look at specific examples rather than to go through the theory. So this is basically going to be sort of a conceptual presentation of Bayesian statistics rather than a computational exercise. So the first example we'll look at is a fairly simple, straightforward one where we look at a diagnostic test, and we're going to look at how you compute predictive values, both positive and negative. Then we'll look at a randomized trial, a published randomized trial of medications for epilepsy. And then the last one, more complicated one, is what's known as probabilistic sensitivity analysis, or sometimes called Bayesian sensitivity analysis. Okay, so as you may or may not recall, when we talked about frequentist statistics, the basic idea here of doing research and then applying statistics to our research problem was that we have a population, which the population, as statisticians define it, includes everybody of interest. And then when we do research, we draw a sample from that population, and we conduct our statistical analysis on the research sample. And so we basically want to know, is our sample representative of the population? And so in terms of a schematic, here's the sort of the idea. In the population, frequentists assume that there's a fixed population parameter. Let's say a mean here, for example. And so we have this fixed parameter, the true population mean. And so we draw a sample from the population, and then we have a mean of our research sample. And basically, what we want to know is this representative of the population. But the frequentist view is based on repeated random sampling, or the possibility of doing that. And so if you were able to draw an infinite number of random samples of size n, whatever that might be, that the mean of those means will equal the population mean. And so from the frequentist perspective, the population parameter, so in this case the mean, is fixed. The sample that you draw, the data that you get, is random. And so you can see that there's a possibility here of what we call random sampling variability, that if we were to draw all these different random samples, the means would not always be exactly the same. And so to sort of illustrate this, I had a population of diastolic blood pressures, of which the mean is 76. And so if I were to draw random samples from this population of n equals 5, or size 5, then I would get what's called a sampling distribution. And so I could arrange, if I arrange the samples that I've drawn, here I have a dozen or so, and calculated the mean of each sample, you can see that there's variability in those means. Now, some of them are really close to the population mean, but some of them are not so close. But we refer to the distribution of means here as a sampling distribution. And so if we calculate confidence intervals from the frequentist perspective, we get depending on what probability you want here, usually it's 95% confidence intervals. So here I have several of my samples that I've drawn. I've calculated the mean, and then I've calculated the standard error of the mean, which, as you may remember, is the variability in a sampling distribution, not the variability in the data. And then I can calculate a lower and upper bound, and so that's my confidence interval for that particular sample. And so in the frequentist viewpoint, from the frequentist viewpoint, the 95% confidence interval means that over repeated sampling, the confidence interval will include the true population mean 95% of the time. And so if I were able to do an infinite number of random samples of size 5, that means a 95% confidence interval means that 95 times out of 100, the interval will include the true population mean, but 5 times out of every 100 samples, it won't. And so you can see here on my little example, I've got a couple here that where the confidence intervals don't include the population mean. And so then we can apply this also to hypothesis testing, where we want to look at the difference, say, in a treatment and control group. And so here I have 2 random samples where the blood pressure is 86 in the control group and 81 in the treatment group. And so my question is this difference of 5 in the diastolic blood pressures, is this difference just due to random sampling variability or chance? And our alternative hypothesis, of course, is that we want to be able to say that this difference is unlikely to have occurred by chance, and so therefore I can reason or make a conclusion that the difference is due to whatever treatment was applied here. And so based on our statistical tests, then we will either reject the null hypothesis, which is to say that it's unlikely that our difference occurred by chance, or we'll fail to reject, which means we really don't have any evidence for a difference. So as I mentioned before, all of our alphas and betas, the alpha error rate, the beta error rate are really arbitrary. And you're probably familiar with the p less than 0.05. That's really a tradition that's come about, and we talked about that to some degree last October. And so if we would happen to set our p-value at 0.01, then a p-value of 0.05 would not be statistically significant, so we would fail to reject the null hypothesis. So you can see that there could be different rejection levels here. And so it's 0.05 doesn't mean that it's necessarily important or meaningful. It just determines what we call statistical significance. And so if the p-value is set at 0.01 and we get a p-value that's less than that, then we reject the null hypothesis. But that means that we would be making a wrong decision 1 out of every 100 random samples. And so our summary then is that p-values are a preset level for rejecting the null hypothesis. It's scaled as a probability from 0 to 1. P-values are not the probability that the null hypothesis is false. It's not the probability that the alternative hypothesis is true, and it certainly is not an index of importance or meaningfulness. And so, as I mentioned, whatever we set our p-value at, our alpha error rate at, then we'll reject the null hypothesis, include that the observed sampling statistics, whether that's a normal distribution or a t or a chi-square, was unlikely to have occurred by random sampling variability. If it's greater than our level, say for example, greater than 0.05, then we'll fail to reject, and the only thing we can conclude is that we found no evidence for a difference. Okay, so to get into then comparing Bayesian and frequentist perspective, as we mentioned, then from the frequentist perspective, probability is the long-run frequency of random events over repeated sampling. And as I mentioned, in this perspective, the data are random, but the population parameter is fixed. From the Bayesian perspective, probability is an index of confidence an investigator has about the occurrence of an event or the value of a statistical parameter. And in the Bayesian perspective, then the data are fixed, but the population parameter is random. And we'll see why that's the case as we go along here. So in Bayesian analysis, we basically have 3 steps. So you have a prior belief about an event, a condition, a diagnosis, a statistical distribution. Then you get new information, which from our perspective here, that's our data that we collect from our sample. And then we revise our prior belief with a posterior or a new belief. And so I'll just give you an example here. Say we have a physician who is seeing a patient. He's prescribed or she's prescribed a particular medication or medications based on their initial diagnosis. But then we get new data from the lab, and so the physician updates the medications based on the current lab results. So that's has a prior event about the medications, then they get new information, the lab data, and then they revise their prior prescriptions for medications based on the new data. And so I use this example because as you then can see or reason that when physicians perform as clinicians, as seeing patients, they really operate as Bayesians because you have a particular diagnosis, you get new information, and then you update the diagnosis. If the physicians happen to be also investigators who are doing research, when they actually do research, then they most of the time become frequentists. So the foundation of Bayesian statistics is the Bayes theorem, which is a conditional probability, the probability of event A given the event B has occurred. And so this formula is well known in its simplest form here. And it comes from Thomas Bayes, who was an English statistician and also a minister. You can see that the Bayesian perspective has been around for a long time, but it wasn't until fairly recently, I think around 1990 or the early 90s, that it became feasible to apply Bayesian statistics to more complex problems, obviously because of the computer processing and computer speed. So my first example here, let me show how this would be applied to a diagnostic test where we have a test that's either positive or negative, and then we have the true state of affairs, which we have listed in the columns of our 2x2 table here. And so the first cell, you have a positive test and you actually have the disease. That's called true positives. And then the second cell here, where you have a positive, you test positive, but you actually don't have the disease, that's called a false positive. And then where you have a negative test, but you actually have the disease, then that's referred to as a false negative. And then the last one, obviously, where you have a negative test and you don't have the disease, then you're a true negative. And so the statistics that we can compute from our 2x2 table here are, first of all, what's known as sensitivity and specificity. And sensitivity is just the number of true positive divided by the number that actually have the disease. And specificity is the number of true negatives by the number of patients that actually don't have the disease. And then we can compute our predictive values, both positive and negative. And so a positive predictive value is the proportion of individuals who have a positive test and actually, or the number of patients who have a positive test, and that's then divided by the sum of those who, all patients who have a positive test. And then the negative predictive value is just similar, that those who have a negative test divided by the number of patients who are actually negative for the disease. So, and we can also calculate the false positive rate and the false negative rate. So in words, then the sensitivity is the proportion of true positives that are correctly identified by the test. And specificity is the proportion of true negatives that are correctly identified by the test. Our positive predictive value is the proportion of patients with a positive test that actually have the disease. And then similarly, the negative predictive value is the proportion of patients with a negative test that don't have the disease. So I have a, well, I'll use an example here to illustrate this, but one of the other important number that we need to know, or that we would hope to have, is the prevalence, or the proportion of a disease that's in a given population. And sensitivity and specificity of a test are independent of prevalence. However, the predictive values, both positive and negative, are dependent on prevalence. And so in terms of applying Bayes' theorem then, and I'm not going to go through the derivation here, but we can use Bayes' theorem for updating our predictive value based on the prevalence, both our positive predictive and our negative predictive value. And so here we would have, this is the probability of disease given a positive test, okay, which is the positive predictive value. And so that's equal to the probability of a positive test given that you have the disease times the prevalence. And then this is divided by the probability of a positive test. So in terms of positive and predictive values, then we can rewrite our formula here, and we can show that the numerator here is equal to the probability of a positive test, but I'm not going to do that right now. So basically then our predictive value is sensitivity times prevalence divided by sensitivity times prevalence plus 1 minus specificity times 1 minus prevalence. So this is the probability of a positive test given that you don't have the disease. So that's 1 minus specificity. Okay, and then you can have a similar argument using the predictive, or for the negative predictive value. So let me use some real actual numbers here. Well, actually they're made up, but so I have a test that's either positive or negative, abnormal or normal. And then the true state of affairs, true pathology here is normal, abnormal, or normal. And so given the numbers that I've put in my 2x2 table here, I can calculate sensitivity, which remember is the number of true positives divided by those who are actually positive for the test. So in this case, it's 231 divided by 258, or 0.895. And then similarly, specificity would be 54 are true negatives divided by the total number of negative for the disease. So that's 0.628 in this case. And then if I calculate my positive predictive value, so that's the number of positives who test positive who actually have the disease. So that's 231 divided by 263. All those, this is all the total number that tests positive. And so that turns out to be 0.878. And then the negative predictive value is 54 divided by the total number of negative tests, in this case 0.667. And my prevalence in my example here is just the total number that have the disease out of, or is the number that have the disease out of my total number that I've tested. So that's, in this case, 0.75. So let's say now that given my information here and I have sensitivity and specificity for this particular test, what if the prevalence was not 0.75, but what if the prevalence was only 0.5? So then I just need to plug that value into my formula. And you see that in this case, it's the sensitivity times my new prevalence. And then the numerator is the same as before. Sensitivity times my new prevalence plus 1 minus the specificity times 1 minus the prevalence. And you can see that the positive predictive value here turns out to be only 0.7. And then if I carry this another step further and solve for the situation where the prevalence is only 0.25, then I get a positive predictive value of only about 0.4. So you can see that the predictive values really depend on the prevalence. And if you have a particular test where you know the sensitivity and specificity, then you, if you apply that test in, say, a different location, a different geographic area or whatever, where the prevalence is different, then you can calculate what the predictive values are going to be. So that's a fairly straightforward, simple example of the application of Bayes' theorem. Okay, so my next example here is actually a published study on the randomized trial of 3 medications for epilepsy. And so I want to go through this in some detail. So I have their statements of their methods and their statistical analysis here to show you how Bayesian statistics is applied in this case. So they do a randomized study. Actually, it's an adaptive trial. I'll explain what that is in a second, in a minute or two. And so they want to compare the efficacy and safety of 3 anticonvulsive agents, levetiracetam, forspheniton, and valproate. Not sure that's how you actually pronounce them, but anyway, they've got those 3 drugs that they want to test and look at the efficacy and the safety of those. So they're going to look at posterior probabilities here. So we know we're going to do a Bayesian analysis in this case. And so the primary outcome well, we're going to look at, but the primary outcome was the absence of clinically apparent seizures and improving responsiveness at 60 minutes after start of the trial drug and without adding any other medications. Then they also looked at safety outcomes, although I believe, as I recall, they analyze their safety outcomes with frequentist statistics. But anyway, so their outcome is basically a yes or no outcome, where it's the absence of seizures and improved responsiveness. Okay, so they used what's called a response adaptive comparative effectiveness design. And so initially, patients were randomly assigned to receive one of the 3 trial drugs. So initially, they're randomized in a 1-to-1-to-1 ratio. In other words, they've got equal numbers in each group. However, after 300 patients were assigned to a particular treatment group, then the notion of response adaptiveness randomization was done on the basis of what the outcomes were in those first 300 patients. So in other words, it's the way that they're randomized after 300 patients may not necessarily be in a 1-to-1-to-1 ratio. And then they did this after 400 patients, 500, 600, and then up to 700 patients. And then they had an early stopping criteria here for either success or futility. So I'm not going to go into the specifics of those response adaptive designs, but that's basically what they're doing, is they're changing the randomization ratio based on the results they get. Okay, so after each interim analysis, the randomization assignment probabilities were updated, okay? And then they were also, the randomization was also stratified by age. And then the key here is, before the assessment of the trial results, all 3 drugs were considered to be equally likely to be the most effective or the least effective. And so here is our prior assumption that all of the, or our prior belief that that all of the drugs were to be equally effective. So they go on to say then that the percentage of patients with treatment success in each group was calculated starting with a uniform prior probability, a uniform 0 to 1. That is, it can take on any value between 0 and 1. And so this is their prior probability distribution, or their prior assumption here. And then they're going to update this on the basis of the observed binomial data and use a beta binomial model or distribution. So the this beta binomial distribution then is their posterior distribution. So the uniform distribution is what's referred to as a non-informative prior, and basically you're assuming that in this case all the groups are equal. So if we were to do a bar graph for the 3 drugs and look at the percentage or the proportion of patients with treatment success, then they would all be equal. They'd all look like that. So what they, in their methods and the way that they analyzed their data and updated their prior to, with the posterior is that they randomly and repeatedly a million iterations, drew from these 3 posterior probabilities to calculate the probability that a given treatment was better than the other 2. And so their definition here of better or most effective was a probability greater than 0.975. So in other words, any one particular treatment we're going to have a threshold here of 0.975 to declare it better than the other. And so here they use a reference in a sense to frequentist statistics, that this would be analogous to an alpha or p-value of 0.025 in a one-sided comparison. And then they give reasons for choosing that threshold. Okay, so the idea here is that when you have your prior distribution, you get your data, then you're going to update your prior from a posterior distribution, but we do that through simulation. That is, we draw a lot of, or a very large number of simulations from the distributions. Okay, so they then define what they mean by an improved treatment or better treatment. That is, that the response rate would be 65%, and the response rate in the other two groups would only be 50%. So they're looking at a 15% difference here, defining, or is the effect size defining treatment success. And then they're going to report the percentage response in each treatment group with 95% credible intervals. And so we're going to talk about that. But in Bayesian statistics, when you do an interval, it's called a credible interval. In frequentist statistics, it's called a confidence interval. And then they mentioned that it was based on the intention to treat population. That is, even if a patient was randomly assigned to get one drug, but they didn't get that or got something else, they're still going to be analyzed as if they were in that particular drug group. Okay, so I did this little illustration here to show you what it would look like if they were, if they showed that there was, that the probability was 0.975, that one group would be superior by at least 15% to the other two groups. And so here is your three posterior distributions. This one, the red one, shows we have our 2.5%. So there can't be, what that means is there can't be more than a 2.5% overlap between the distribution that's better and the other two distributions that aren't as good. And so then the rest of this area here in the red one would be 97.5%. Okay, or 0.975 probability. So that's what if their hypothesis was supported, that's what it would look like. And I did this because this is how they present their data, their results. You can see here that they plot then the posterior distributions for each drug. And you can see that in contrast to the one that I had before, there's virtually no difference in the distributions. And so for each drug here, we have a 95% credible interval. And so remember now that what a credible interval means, that there's a 95, the probability is 0.95, that the drug percent of success is in this interval, lies in this interval. If this were a frequentist interval, what it would mean is that 95 times out of every 100 random samples, the percent of success for that group would fall in that interval. So that's, you can see that those are different definitions. They're in a sense trying to answer a similar idea here, a similar question, but the interpretation of intervals, confidence or credible intervals, is quite different. And so you can see here that they conclude that there's no significant difference between the groups because they're virtually identical, and the confidence intervals pretty much overlap each other. And then the picture of their distributions are, the graph of their posterior distributions are, again, virtually identical. So that's using the Bayesian statistics to do this analysis. which could also, it's not that complicated. It could have also been done fairly easily using frequentist statistics. So again, just to reiterate here, when we talk about a Bayesian interval, credible interval, we mean that there's a 95% probability that the interval includes the population parameter. And from a frequentist standpoint, the confidence interval means that over repeated sampling, the interval will include the population parameter 95% of the time. Okay, so then my last example here, which is the most complicated one of all, is probabilistic sensitivity analysis, or it's sometimes referred to as Bayesian sensitivity analysis. And so we're using this as an example of a quite complicated design and to answer a fairly complicated question. And so we're doing this when we have multiple input variables to estimate multiple parameters. So in the last example of the epilepsy study, there was just one parameter that we wanted to estimate, and that was the percent success. And we do this because estimation of a parameter is sensitive or may be sensitive to changes in the input variables. And also in this case, an example I'm going to use, our prior and posterior distributions, whatever they might be, are the same for a given variable, but we might have different distributions for other variables, okay? So the example I'm going to use is I'm going to apply this probabilistic sensitivity analysis to cost-effective analysis. And so in a cost-effective analysis, what we want to do is compare the cost and the effectiveness of two, or we can do more, treatments. And the data that we have are patient-level costs, and then some measure of how effective the treatment is. So we have a particular outcome, which is in some form basically survival. That is, how long patients live given the treatment or no treatment. And what we calculate is what's called life years gained. In other words, what is the gain in life years that you would normally expect given, say, age, gender, whatever other variables you might want to put in there as a result of the treatment. And in most cost effectiveness studies, this is translated to what we call quality adjusted life years, or QALYs. And they're calculated by multiplying life years gained by a utility, which is a scale from 0 to 1. It's a measure of health status where 0 means death and 1 means perfect health. And so usually you can get a utility value for each patient several ways. Many studies actually have a measure where like a patient reported outcome where they rate their health status before or after their treatment. Sometimes if you don't have that on a patient level basis, you have to use other databases where they give average utilities given age, sex, whatever other demographics or comorbidities you might want to include in that. And then the other component that we need is the cost of the treatments. And so costs might include or would include procedures. So for example, if you have a coronary artery bypass, how much does that procedure cost? You would have physician cost, medications, obviously. You might be hospitalized after the treatment. So what is the cost of that? Additional hospitalizations. You might have office visits or lab tests that would be included in cost. In the past, people have tried to take into account things like, wages lost, time away from work, or things like that. But that's really hard to get a valid measure of. So most of the time, that's just ignored. But those are the two components here that we're going to look at, are quality adjusted life years and then the cost of different treatments. And so here's an example from a recent study that we did where we have a cost per episode. And so you can see here we have for a fatal CV event for different age groups, there's a different mean cost. And then we have a minimum and maximum, and then you have to, in this, you have to specify where you caught, where these costs came from. You can't just pull them out of thin air. So there's always going to be a reference for where you came up with these costs. And so, for example, here for a fatal CV event between 45 and 64 cost $18,940. And then I have another for an example here for a non-fatal MI for the same age range, the cost is $22,542. So when we do our cost-effective analysis and just calculate the cost, what the cost might be, we're going to use the average cost here. But of course, this can vary, as you can see. And so what we do is we specify a distribution, a theoretical statistical distribution that models these data. And so for costs, you can see that we are using a gamma distribution to model these costs. So each one of these for different age groups would have a different gamma distribution. And you say, well, where do you get that from? Well, if you were to look at costs in most any study and do a histogram of it, they would all look something like this. And so you can see that it's obviously not a normal distribution, but it models, or a gamma distribution is probably the best model for this. And the reason is that because you have data that's right skewed, because most of the costs are within a certain range here, but you always have extreme costs. And these are costs that you don't want to leave out of your analysis, so you include them in there. So we are going to model cost as a gamma distribution. Okay, and then we would also have utility values that we would also need to specify how we're getting these, so there's always a reference for where these came from. And so, for example, here we would have a utility value if you had a history of a MI, then utility value would be 0.96. And so then if that's, you had a history of an MI, then your life years gained would be multiplied by 0.96. And so you can see the different, values here, where if you have no history, then you have a value of 1. Then if you actually die during the trial, then if the person dies during the trial, then their utility would be 0. And then you can see that we're going to model this as a beta binomial distribution. That's the same distribution that they had in the epilepsy study. But we're using that because this is a value that can range between 0 and 1. And I'll just mention here about what's called disutilities, which is somewhat controversial, but these have been calculated in some fashion or another, that if you experience one of these during the trial, then your utility value is lessened, or you subtract the disutility value from your utility value. So for example, if you had a history of MI and your utility value is 0.96, if you had an MI, another MI during the study, then you subtract 0.0079 from that. But so that's the two primary distributions that we're going to look at in cost-effective analysis. So the statistic that we're interested in here is what's called the incremental cost-effectiveness ratio, or the ICER. And that's basically the difference in costs between our two treatment groups divided by the difference in efficacy of the two groups. And so the interpretation, so for example, if the ICER came out to be $50,000, then what that means is that we would need to spend $50,000 on our treatment to gain one quality adjusted life year. Okay. So what we do then is we're going to plot our incremental cost effectiveness ratio in what we call the cost effectiveness plane. And so the vertical scale here is the cost, and so as it increases, that's more costly. Our horizontal scale here is effectiveness, our measure of effectiveness. And so it's increasing as we go to the right here. So if we divide this into quadrants, then the first quadrant here in the upper right is what we call the cost effectiveness quadrant. That is, cost is higher for the treatment, but so is effectiveness. In quadrant 2, our treatment is what we call dominant. That is, not only do you have a treatment that's lower cost, but it's also more effective, which is what everybody would like. Our third quadrant is where we have less cost, but effectiveness is also less in the treatment group. And then the sort of the worst possible outcome is quadrant 4, where the treatment is dominated, where not only is the cost higher, but effectiveness is also lower. So what we're going to do is we're going to do our probabilistic sensitivity analysis, and we're going to plot our results in this cost effectiveness plane. So this is kind of a schematic of what we do. This is at a real conceptual level. It's not actually how it's done. I should say it is how it's done. It's just that the computations are fairly complicated. But the idea is that we're going to do random draws, and we're going to do a lot of them from our different distributions here. So for example, we might have a distribution here for the cost of a fatal cardiovascular event. And then we would have a distribution for the cost of having an MI, and then the cost of a stroke. And then so we maybe would also have a distribution here for the cost of a medication. We'd actually have probably a lot of different distributions for different medications. And then over on the right side here, I've plotted the distributions for utility. So if a person has a history of MI, a history of stroke, or these other possibilities. So for example, here we might draw a person has a stroke during the study, so we would draw from this cost distribution. We would draw a utility from this distribution if they had a history of angina. And then if they were on atorvastatin, we would draw from this distribution. So you can see we're drawing from multiple distributions at the same time. And then actually we would have another one here that I haven't shown, and that we would need to have a distribution of life years gained, which could be different for different age groups. So you can see that this is fairly complex, but say our random draw here, we would do say 1 to 100,000 of these, and so we would get, for the two groups, we would get a difference in cost and a difference in qualities, and we'd have 100,000 of those. Okay, so when we run our simulation here, then we come up with a picture that would look something like this, where we do a contour plot, where as we have the mean cost difference on the vertical axis and the mean effective difference on the effectiveness difference on the horizontal axis. And so each of the dot represents one of the simulation draws from our different distributions. And then the inner ellipse here is 90% of all of the simulations fall within this ellipse. The second one here is the 95% ellipse, and then the outer one would be the 99% ellipse. So 99% of all the simulations fall within the outer ellipse. And then I've drawn the, typically what you do is you have the horizontal and the vertical axes at 0. We don't have 0 on the cost here because none of them are negative cost. The treatment group in this case always costs more. But you can see that here there's really not much difference, that there's no real advantage here of the treatment group in terms of cost effectiveness. So there's a lot of the simulations fall in quadrant 1 here, but a lot of them also fall in quadrant 4. So this is sort of what the results look like, but we would also have tables with actual numbers in them. So this is how Bayesian analysis can be used for quite complex situations that are not as easy to do with frequentist statistics. So in summary then, our frequentist position, remember, is based on repeated sampling of data from a population. And the confidence interval means that over repeated sampling, the interval will include the population parameter some percent of the time, whether it's 95, 99%, or 90%. In the Bayesian perspective, we have a prior distribution. Then we collect our data, and then based on that data, we update our prior distribution with the posterior distribution. And then a Bayesian credible interval means that there's a particular probability that the interval includes the population parameter, like a 95.95 probability or 0.99. Okay, so then frequentist methods are used for what I've called simple or straightforward analyses. The analyses may not be simple. You can do like a mixed effects model, which is kind of complicated, but in some cases it's just easier to do that. But the advantage of Bayesian methods is they can handle complex multi-parameter analyses that are difficult for frequentist methods. And when you do a Bayesian simulation, usually like the first 10,000 to 50,000 iterations are what's referred to as burn-ins. And that's because your model may not be stable initially when you first start to run simulations. So you use, after the burn-in period is then the simulations you use for your distribution model. As far as software goes, you can do Bayesian statistics in some of the major packages. The packages, though, that are used probably primarily for Bayesian analyses are WinBugs or OpenBugs, which I believe OpenBugs is a more user-friendly version of WinBugs. But basically, these are programs that will do the large number of simulations that you need to do in Bayesian analysis. So you can see the difference here, as I mentioned, between Bayesian and frequentist is that in frequent analysis, we're operating on the actual data as far as doing our statistical test, or statistical model. In Bayesian statistics, we're using theoretical distributions, theoretical statistical distributions to model the parameters or whatever it is that differences that we're interested in. So I've have just a few references here. The first one is the epilepsy study that we looked at, if you're interested in looking at the details. The second one is sort of a, I guess, a kind of an introductory presentation of Bayesian data analysis, if you're interested in getting more into it. And then the last reference I have here is an interesting comparison of credible interviews and confidence intervals that is, like I say, is kind of interesting in comparison, comparing the two in terms of what they represent and what they mean. So I want to again thank you for your attention, and I don't know, we're getting close to being out of time here, but if there's any questions, we can use the Q&A, or if you just would rather email me any questions about Bayesian statistics. Actually, could be a number of you out there that know more about Bayesian statistics than I do. But anyway, if you're interested, just send me an email. So again, thank you for your time and attention, and we will again meet next month in March. I believe it would be March 19th. Take care and have a good day. Oh, let's see. Okay, we have one question here. Okay. Okay, so the question is, are the draws from the same patient or different patients? Okay, the draws are not from patients, they're from distributions. And that's, I say, the difference between Frequentist and Bayesian statistics, that you're proposing that some prior model from for a particular statistic or model, and then you use your data to update that model or that particular distribution. So in the epilepsy example, they assumed a uniform distribution to be in their prior distribution. In other words, they were assuming that all treatment groups were equal, and then they used a beta binomial distribution, or that was that was updated then with a beta binomial distribution from their data. So in answer to your question, that's in Bayesian statistics, you're not drawing the same patients over and over again. You're drawing from theoretical statistical distributions. Okay. Okay, if there are no other questions, I don't think. Okay, again, thank you. Oh, let's see, maybe there are. Let me see here. Okay. Oh, okay. Another question is, could you also talk a little bit about bootstrapping? Okay, so bootstrapping is then applied to the actual data. So that's a Frequentist method. So you draw random samples from the data with replacement, and you do that a large number of times. So again, the difference with the Bayesian perspective is you're making random draws from distributions. Okay, let's see. Then another question is, oh, okay, sure. The question about there is a recording. I think Georgetown will send that out. And then sure, you can email me any questions that you have. Okay, then another question asks, are you saying that in the epilepsy example, they analyzed and interpreted the data correctly, but wrote their methods incorrectly? No, they wrote their methods correctly as far as the Bayesian part of it. There were some others, I didn't go into that, where they actually use frequentist statistics, but I use this for a Bayesian example. So their write-up was correct, although they still use the significance or not significance, which, as you may or may not remember from our very first presentation, the American Statistical Association doesn't want to use those terms anymore. Let's see. Do we have any others? Okay, I think that's. Oh, okay. So there's also when to know if you have the slides. Sure, I can put the slides out to the folks at Georgetown, and then I think they can probably put them on somewhere where you can then access them. Okay, great. I think that's our time, so I'm going to stop sharing and sign off. Again, thank you for your